Our laboratories are equipped with board technologies which allow us to achieve our goal, excellence in research. We show the most important and newest part of our facilities and part of our research. Notable part of them have been started during the last 2 years. We would like to highlight continuous improving of our research quality. The Smart Technologies Division of the Biotechnology Scientific Laboratory was established on October 9, 2019. The division provides modern research infrastructure for sciences of all LLU faculties and institutes. In LLU, BZL, and BMN, it is possible to perform microbiological and mycological testing of both plant and animal samples, as well as soil improvers in accordance with the requirements of ICO and LVC standards. In the laboratory, it is possible to determine the antibiotic resistance of bacteria isolates with the classical method, which offers the sample owner of the opportunity to choose the antibiotic to be used in the further treatment. Highly accurate and fast identification of bacteria and microscopic fungi isolated from tested samples is available in the laboratory with the automated MALDI-TOF mass spectrometer, which allows identification of bacteria grown on selective and chromogenic media. Identification of isolated bacteria with a VITEC MS automated mass spectrometer, at the same time determination of MIC of the antibiogram. Samples of biological material, blood and DNA of various farm animals, cows, horses, sheep, pigs, and goats are stored and supplanted in the gene bank of Department of Molecular Biology and Microbiology. The aim is to identify and study local engagement varieties in order to conserve genetic resources. Local endangered varieties for our national wealth, and our task is to promote the cultivation of these varieties in order to ensure the preservation of genetic diversity in our country. LLU, BZL, and BMN has the possibility to determine the amount of E. coli, coliforms, and enterococci in all types of water, surface and drinking water, by accredited methods. Sequencing of DNA fragments, e.g. identification of bacteria based on 16S RNA gene sequencing results. Fragments analysis, e.g. for paternity and maternity in dogs. Extraction of DNA from milk samples. Designed to determine the quantity and quality of DNA. For accurate and rapid identification of the samples to be determined. Research on organogenetic resources to improve the international descriptors and to characterize the Latvian organogenetic resources. The research will be a part of Irina Savitska's doctoral thesis. The device has been used to create high-resolution images of surface by scanning with a focused electron beam. Studies on biological objects, materials, surfaces, and structures. Measurements of materials of different origins. Evaluation of structural homogeneity, pores, inclusions, crystals. Raman spectrometer has been used in cell research and visualization of intracellular molecular components, real-time dynamics research. Chromatography mass spectrometry system is used for the qualitative and quantitative analysis of the chemical composition in a wide range of materials and samples. Identification databases are available for the pesticide and veterinary drug residue determination. It incorporates a supercritical carbon dioxide extraction device which is in conjunction with a liquid chromatograph and a mass spectrometer and makes it possible to fully automate the analysis process. The Department for Agronomic Analysis started its activity in 1995 as a scientific laboratory of agronomic analysis in the LLU and has been accredited since 1999. The areas of activity of the department are physical and physico-chemical testing of samples of plant and animal origin, as well as soil improvers. The Department of Agronomic Analysis offers to determine various physico-chemical parameters, both by chemical methods and NIR, near-infrared light spectrometers, as well as to perform accredited analysis of physico-chemical parameters and soil improvers. Determination of starch and phosphorus content in animal feed, measuring the cutting angle of polar active substances. Preparation of samples for determination of crude protein content in animal feed. Combustion systems are suitable for heating the sample into stable tubes for determine the total protein content of the sample according to the Giedel-Nitrogen method. Determination of the nitrogen total protein, bound protein content of the sample according to the Giedel method. Determination of crude fiber content and lignin content of animal feeding stuffs. Determination of the fiber content of acid containing detergents. Determination of amylase, treated and soluble fiber in feeding stuff. Determination of fat content in animal nutrition. Identification and quantification of volatile compounds in a sample. The device is designed for fatty acids, cholesterol, esters, aldehydes, and etc. for the determination of compounds in food. The Biochrom 30 Plus is designed for the quantitative and qualitative analysis of free amino acids in physiological fluids. The Biochrom 30 Plus system can identify and quantify up to 56 amino acids and derivatives found in various psychological sample matrices. Determination of carbohydrates, fructose, sucrose, lactose, glucose in samples is used in scientific research work determining the content of various elements in biological materials of various origins: soil, plant, water, sediments, product of animal origin, and etc. In cooperation with leading scientific institutes in Europe, races of pathogens were studied. Laboratory of plant pathology is provided with all necessary facilities to study fungal pathogens and their biological traits. The aim of the project is to identify species of Botrytis and clarify their morphological and genetical diversity. The aim of the project is to describe pathogen diversity, sensitivity to fungicides, and precise life cycle of disease. In our laboratory, botrytis cinerea resistance against fungicides were made. Studies are based on assessment of fungal colony area. The other field of our expertise is to identify the Fusarium species and fungiform genera Pyranophoria, which infect out in Latvia more frequently in the last years, to find the best solution of the plant protection methods. The molecular and morphological methods has been applied. The equipment is used to determine soil respiration rate, to assess the growth and physiological activity of microorganisms in different substrates, to access gas exchange and biodegradation processes. Under the Latvian Ministry of Agriculture funded project identification of bees, Apoidea, caught in various agrocenes is being made. Wheat seed germination was tested to assess the degree of seed dormancy. Different weed management methods are being developed, including high precision of mechanical weeding and laser treatment. Biointegration of a new type woven aortic prosthetic from a composite material in a dog abdominal aorta. Research and technological solutions for sustainable sea buckthorn cultivation are full use. Sea buckthorn is feed supplement in calves. The effect of Lactobacillus fermentum on the health of dairy cows during the milk yield increased period for the prevention of SARA. Appropriate laying head breeds evaluation for commercial production for organically reared chicken eggs in Latvia using different feeds. Testing potassium humate extracted from Latvia black peat as a new feed ingredient in dairy cows. Use of stem cells in the treatment of degenerative disease of the dog's elbow joint. Preservation of cow genetic resource in Latvia using embryo transfer and related biotechnologies. Various high-resolution microscopes with the possibility to digitize the image, as well as to process it with the special computer programs are used for the examination of histological samples. The program provides quantitative measurements that are essential for statistical data processing. Quantitative measurements include microstructure counting, length, width, angle, and area measurements, as well as marking of required structures. For example, an intermicroscope network with cameras makes it possible to digitize an image, as well as process images with a special computer program and project a histological image on the wall monitor. The central microscope is connected to 12 more microscopes and a network of monitors, which allows to project a histological image from any microscope to other microscope monitors. Histological examination is applicable to various veterinary fields, with the aim of determining the cause of disease, pathogenesis, and morphological changes. The flow cytometer is capable of immediately examining large numbers of living cells and determining cells phenotyping and differentiation into subpopulations. The device is used in many fields: immunology, oncology, stem cell research, and etc. Start using a flow cytometer in the veterinary immunology field to evaluate calf lymphocyte subpopulation in the first month of life after feeding with a specially developed feed additive based on the sea buckthorn extract. The instrument ensures gentle transfer of living cells to the slide without loss of material, used for analysis of biological material in the field of cytology and in the cell culture studies, including stem cell studies. Morphological studies of animal carcasses begin in the necropsy hall, which is equipped with special equipment that allows work with various types of samples, including high-risk material. The equipment also makes it possible to examine the internal structure of the bones, spinal cord, brain, nasal cavity, etc. A pathological examination shall be carried out to determine the cause of the disease or death of the animal. For the morphological examination of tissues and cells, thinly cut fixed tissue samples are prepared. The histological cutting table is equipped with vertical and horizontal ventilation flow to maximally protect workers and the environment from toxic fumes and chemicals. The tissue processors provide dehydration and lipid purification of the fixed tissue sample and prepares the sample for further processing. The equipment is of vacuum enclosed type, which ensures high-quality sample preparation. The sample is placed in paraffin block using a paraffin pouring station. The prepared paraffin block with the tissue sample is cut very thinly with the help of microtome. The microtome is fully automated with programming capabilities and is designed for cutting paraffin blocks of various sizes. The microtome can cut tissue samples up to 3 micron thin. Transfer the cut sample of the surface of the water for further application to the slide and staining. Cell and tissue changes are assessed under a high-resolution microscope. Multiplayer researchers, up to 10 people simultaneously, can examine a single sample, perform parallel testing, and participate in the discussion of the analysis of the study material. A network of meteorological stations has been set up to provide accurate meteorological data needed for field experiments. The stations are equipped with various sensors, including additional wireless sensors, to capture microclimate differences. Meteorological data are used to provide farmers with forecasts that are important in integrated farming for the rational use of pesticides, as well as to work on new prediction models. Research on tomato diseases, effectiveness of pollinators, and activity of natural and vertebrate predators is carried out in collaboration with industry partners in our greenhouse, growing tomatoes hydroponically and using different light wavelengths to increase the yield and quality of it. The personnel of the Institute of Soil and Plant Sciences has the opportunity to conduct research on plant growth, development and crop formation in phyto chambers, greenhouses, and vegetation rooms. The obtained data have been and are used for the implementations of projects of UMAL and IRIS. At the Institute, it is possible to determine the reflection and absorption spectra of plants' leaves. These data allow to assess the physiological status of plants in a non-destructive manner, thus updating data on the physiological status of plants and crop formation by time-saving methods. Excellent and various possibilities for different field trials are available. We have strong cooperation with other sciences. For example, glasshouse gas emissions were measured depending on agronomic practice. The farm is equipped with modern technologies and machinery, and staff have experienced knowledge and competence related to conduction and maintenance of field trials. Focus area of forest research is climate-smart forestry. forest adaptation and ecosystem services in the context of long-term production forestry that is an important part of national economy. Project will provide a reliable information on financial and impact of windstorms for forest owners and policymakers. Results of project will cover the knowledge gaps in the primary wind dams for the main species and age covering private forests. It is also including assessments of impact of a lower wind speeds when the tree is not snapped, but structural wood damages has occurred, now visible from the surface of the tree. To measure this effect, sap flow monitoring is carried out using HFD sensors. Analysis of a climate-growth relationship, including for an emerald species, predicted to be suitable for the conditions in Latvia in the near future due to climate change, is carried out in close cooperation with Latvian State Forest Research Institute Silava. Novel interdisciplinary approach is used in the project for studying which ecological values and which tools of nature conservation are necessary to ensure efficient protection of biodiversity in the context of requirements for certification of forest management, as well as social acceptance of adaptive forest management and measures important for long-term reductions of risk and vulnerabilities of forest stands towards different natural disturbances. Close cooperations with Latvia State Forest and the research is developed.